The ninth recent discovery on the list is that the junk DNA paradigm has been shown to be false. In this video, I will recount the rise and fall of the junk DNA paradigm. This is important to the debate about design because diametrically opposed predictions about functionless DNA in the human genome had been made from the perspectives of random mutation and natural selection versus intelligent design. The predictions of the design perspective turned out to be correct. I want to acknowledge Casey Luskin at the Discovery Institute for help with much of the content of this video. This slide shows a timeline for some important events related to this topic. References claiming that a large fraction of our DNA is junk or garbage date back to the early 1970s. The concept of DNA as selfish became popular in the late 70s upon the publication of a book by Richard Dawkins. This is the gene-centric view of evolution, the hypothesis that genes are selfish replicators that use organisms to protect and replicate them. This hypothesis also predicted that much non-functional junk would accumulate in genomes. The project to sequence the human genome ran from 1990 to the early 2000s. In the middle of that decade, Francis Collins, who led the government-funded sequencing project, stated that roughly 50% of the genome is non-functional. The ENCODE project, a large project that the government funded following the Human Genome Project, was critically important for this topic. It started in 2003 and is still ongoing. This project's goal is to identify all the functional elements in the human and mouse genomes. In 2012, a suite of papers were published from this project stating that the vast majority of our genome, more than 80%, is functional. In 2021, a paper in Genome Biology and Evolution stated that the days of junk DNA are over. The Human Genome Project revealed that only a few % of our DNA codes for protein, which reinforced the thinking of many scientists that most of our DNA was non-functional junk. But the ENCODE project brought about a major revolution within biology because it showed that most of our DNA was transcribed into RNAs that do not code for protein but seem to be doing something else. Subsequent work has shown that those RNAs are involved with regulating the transcription and translation of genes. So coding for proteins, as shown on this slide, is only a minor part of what RNA does. A whole new world, so to speak, of RNA function has been uncovered. This paper from 1972 may be the earliest to claim that a large fraction of our DNA is junk or garbage. The Selfish Gene by Richard Dawkins was published in 1976. In a poll in 2017, this book was voted as the most influential science book of all time. In this book, Dawkins argued that we are survival machines, robot vehicles that are blindly programmed to preserve the selfish molecules known as genes. He went on to write, "The simplest way to explain the surplus DNA is to suppose that it's a parasite, or at best a harmless but useless passenger, hitching a ride in the survival machines created by the other DNA. The accumulation of surplus DNA was expected based on the idea that natural selection does not remove nearly neutral recessive mutations. Mutations that have no significant effect on fitness will persist in the genome, and so useless junk eventually accumulates. This article from 1980 by W. Ford Doolittle and his co-author supported the concept of selfish genes and the prediction of an accumulation of non-functional DNA. This article from 1980 by Leslie Orgel and Francis Crick also supported this hypothesis. Some advocates of intelligent design raised red flags early on about assuming that most of the human genome is useless junk. In 1994, Forrest Mims, a scientist with the perspective of intelligent design, submitted a letter to the journal Science, warning against assuming that the majority of the genome would be junk. In 1998, William Dembski wrote that, "On an evolutionary perspective, we expect a lot of useless DNA. If, on the other hand, organisms are designed, we expect DNA as much as possible to exhibit function." And indeed, the most recent findings suggest that designating DNA as junk merely cloaks our current lack of knowledge about function. Design encourages scientists to look for function where evolution discourages it. The belief that most of our DNA was junk persisted. In 2006, in his book, The Language of God, Francis Collins, then director of NIH, wrote, "Mammalian genomes are littered with repetitive DNA, with roughly 45% of the human genome made up of such genetic flotsam and jetsam." In 2008, Dennis Alexander wrote that 50% of our genomes are composed of non-functional parasitic DNA. A closely related statement was included in a 2013 textbook called Evolution: Making Sense of Life. It says, over half of the genome is composed of neither genes nor vestiges of human genes nor regulatory regions. Instead, it's made up of parasite-like segments of DNA. In contrast, a few years earlier, in 2011, ID advocate Jonathan Wells published a book titled The Myth of Junk DNA. He cited evidence from genomic studies that non-protein coding DNA performs a wide variety of important functions. He also cited the fact that many human diseases correlate with changes in non-coding DNA. The ENCODE project was a massive effort to build a comprehensive parts list of functional elements in the human genome. The participating institutions in the consortium are shown in this map. ENCODE stands for Encyclopedia of DNA Elements. In 2012, a series of papers were published from the ENCODE project reporting their initial results. The lead paper in Nature stated, "The vast majority, 80.4%, of the human genome participates in at least one biochemical RNA and or chromatin-associated event in at least one cell type." Another research paper published in Science stated, "When researchers first sequenced the human genome, they were astonished by how few traditional genes encoding proteins were scattered among those 3 billion DNA bases. Instead of the expected 100,000 or more genes, the initial analyses found about 35,000, and that number has since been whittled down to about 21,000. In between were megabases of junk, or so it seemed. This week, 30 research papers, including 6 in Nature, and additional papers published by Science, sound the death knell for the idea that our DNA is mostly littered with useless bases. This paper, published at the same time as the others, stated that the 80% number is likely to go higher because the analyses for functional elements only included 147 cell types, whereas the human body has a few thousand cell types. They state, a given part of the genome might control a gene in one cell type but not others. If all the cell types are included, functions may emerge for the phantom proportion. It's likely that the 80% will go to 100%. This image illustrates how the functional assays within the ENCODE project address different potential functional roles for non-coding DNA in the regulation of gene expression. The regulation of gene expression in different tissues is tremendously complicated. There are many different ways that non-coding DNA can influence the expression of a gene. Portions of DNA that are far away from a gene, as well as portions that are immediately adjacent to a gene, can affect the expression of the gene. Also, recall that DNA is packaged into a structure called chromatin, and the alteration of the chromatin structure and the packaging or unpackaging of the DNA within chromatin affects gene expression. I won't go into all the details here. This image shows that one of the assays of the ENCODE project was to measure how much of the DNA was transcribed into RNA. They found that the vast majority of the non-coding DNA is transcribed into RNA, at least implying a function. This is an illustration of regulation of gene expression from a popular cell biology textbook. There are 2 important points to note from this slide. First, it shows that portions of DNA that are far away from a gene, as well as portions that are immediately adjacent to a gene, can affect the expression of the gene. Second, this slide also shows that early on it was believed that proteins were thought to be the main regulators of gene expression. The colored objects attached to the DNA strand in this image are all proteins called transcription factors. The ENCODE project and subsequent work has shown that while proteins are involved, the main regulators of gene expression are non-coding RNAs. Eric Green, director of the National Human Genome Research Institute, the institute that directed this project, stated, during the early debates about the Human Genome Project, researchers had predicted that only a few % of the human genome sequence encoded proteins, the workhorses of the cell, and that the rest was junk. We now know that this conclusion was wrong. He went on to say, ENCODE has revealed that most of the human genome is involved in the complex molecular choreography required for converting genetic information into living cells and organisms. He further stated that by carefully piecing together a simply staggering variety of data, we've shown that the human genome is simply alive with switches, turning our genes on and off, and controlling when and where proteins are produced. ENCODE has taken our knowledge of the genome to the next level, and all of that knowledge is being shared openly. Next, I'll discuss the reactions of several scientists to this discovery, because this is highly relevant to the debate about design. First, I'll start with Richard Dawkins. This slide contains some quotes from him before the ENCODE project reported its results. As I mentioned earlier, in 1976 he wrote, "The true purpose of DNA is to survive, no more and no less. The simplest way to explain the surplus DNA is to suppose that it is a parasite." In 2004, he said, "Creationists might spend some earnest time speculating on why the Creator should bother to litter genomes with untranslated pseudogenes and junk tandem repeat DNA." In 2009, he wrote, "The greater part, 95% in the case of humans, of the genome might as well not be there for all the difference it makes." This slide contains statements by Richard Dawkins after the results of the ENCODE project were reported. He said, "There are some creationists who are jumping on ENCODE because they think it's awkward for Darwinism. Quite the contrary, of course. It's exactly what a Darwinist would hope for, to find usefulness in the living world." He went on to say, "We thought that only a minority of the genome was doing something, namely, that minority which actually codes for protein. And now we find that actually the majority of it is doing something." Further, he states, "The rest of the genome, which had previously been written off as junk, is now understood as the program that's calling into action the protein-coding genes." So after several decades of claiming that non-functional junk DNA is an expected consequence of natural selection, Dawkins's reaction to the results of ENCODE was to claim that function for non-coding DNA is exactly what a Darwinist would expect. More importantly, there was a very strong reaction by a group of evolutionary biologists to the ENCODE project findings. This was expressed in a sharply worded article stating that the project's findings could not possibly be correct. This group reached that conclusion by reasoning from within the traditional gene-centric neo-Darwinian framework. I will review this controversy in some detail because it's highly informative and relevant to the debate about design. In 2013, this group published the paper shown here to state their case. Notice the unscientific and sneering title. I'll come back to that shortly. They believed that the conclusion of ENCODE, that the majority of our DNA is functional, could not possibly be correct, branding it absurd in the abstract. A main aspect of their argument was the definition of the word function in biology. They stated that function in biology must be defined within a strict Darwinian interpretation, meaning that DNA elements are functional if and only if they are maintained by selection. Simple calculations from population genetics, which I'll go over below, indicated that only about 10% of the human genome could be functional, meaning maintained by natural selection. Therefore, they argued that it was not possible that 80% of the human genome could have function, defined in this way. They strongly criticized the ENCODE researchers for determining function experimentally using an evolution-free definition of functionality. They argued that unless a genomic functionality is actively protected by selection, it will accumulate deleterious mutations and cease to be functional. The absurd alternative, which unfortunately was adopted by ENCODE, is to assume that no deleterious mutations can ever occur in the regions they have deemed to be functional. Such an assumption is akin to claiming that a television set left on unattended will still be in working condition after 1,000,000 years because no natural events, such as rust, erosion, static electricity, and earthquakes, can affect it. Hence the title of their paper. The logic in the above paragraph, derived from a particular view of evolution, may be difficult to follow because, in fact, ENCODE did not assume anything. They simply used experiments to test for function. Further in the article, the authors state, "We urge biologists not to be afraid of junk DNA. The only people that should be afraid are those claiming that natural processes are insufficient to explain life, and that evolutionary theory should be supplemented or supplanted by an intelligent designer," with references to Dembski and Wells. ENCODE's take-home message is that everything has a function and implies purpose. And purpose is the only thing that evolution cannot provide. So it's very clear that something fundamental to evolutionary biology was at stake. They recognized that the question of the presence of junk DNA in the human genome directly impacted the debate about intelligent design. Junk DNA is not only expected by, but is demanded by the view that the genome came about by and is maintained by mutation and selection. If you're interested in this topic, I strongly recommend reading the entire article, but I won't quote from it any further here. Science writer and former editor of the journal Nature, Philip Ball, wrote a book in 2023 about the revolution that has taken place in biology. He wrote this about the controversy over the ENCODE results. He wrote, "But some biologists didn't just disagree with what the ENCODE team was saying, they hated it. When a finding excites passions to this degree, you know that cherished ideas are being threatened." The emphasis on the word hated is in the original text. Ball further wrote, "What seemed to be at stake in these arguments was the very integrity of biology itself, specifically the need to divorce it from purpose." Ball is not an advocate of intelligent design, but he has come to the conclusion that it's now unavoidable to include purpose in our conception of biology. He goes on to say, "To accuse an internationally renowned team of scientists of opening the door to intelligent design is akin to an ideological accusation of a betrayal of the faith." Dan Grauer, the lead author on the paper that I just mentioned, and a professor in the biology department at the University of Houston, stated in a lecture at that university, "If the human genome is indeed devoid of junk DNA, as implied by the ENCODE project, then a long, undirected evolutionary process cannot explain the human genome. If, on the other hand, organisms are designed, then all DNA, or as much as possible, is expected to exhibit function. If ENCODE is right, then evolution is wrong." An article in The Guardian described this controversy within science. That article includes a disparaging statement by Dan Grauer about the enormous team of researchers from many top research institutions around the U.S. It reads, "Everything that ENCODE claims is wrong. Their statistics are horrible for a start." The lead author of the paper, Professor Dan Grauer of Houston University, Texas, told The Observer, "This is not the work of scientists. This is the work of a group of badly trained technicians." I'll explain the reasoning of Grauer in some detail, so the basis of his strong belief about this is clear. His reasoning was spelled out in detail in another paper in Genome Biology and Evolution in 2017. His reasoning is based on the genetic load argument. This says that a species cannot survive if the average number of harmful mutations to functional DNA per generation exceeds the replacement fertility rate per person, which Grauer chose to be 1.8 or 3.6 per couple. Basically, the rate of getting rid of harmful mutations must be at least as great as the rate of generation of harmful mutations, or a species cannot survive. The term harmful implies that the mutations are impacting functional, that is, selected, portions of DNA. Given the known genome size and mutation rate, and a reasonable estimate of the fraction of mutations that are harmful, such as about 10%, then according to this calculation, if greater than 10 to 15% of our DNA were functional, harmful mutations would accumulate and our species couldn't survive. The calculation goes like this. On average, each human has about 100 mutations in their DNA that did not exist in the genomes of the parents. About 10% of these mutations are expected to be harmful if they occur in functional regions of the DNA. So if 100% of our DNA were functional, then the rate of harmful mutations per individual would be 100 times 0.1 times 1, which equals 10. This value of harmful mutations per individual is far greater than the fertility replacement rate of 1.8. And so if that were the case, our species couldn't survive. If only 10% of our genome is functional, then the rate of harmful mutations per individual would be 100 times 0.1 times 0.1, which equals 1, which is close to the limit. If only the 2% of our DNA that codes for proteins is functional, then the rate of harmful mutations per individual would be 0.2. This value is sufficiently low that it's consistent with our survival as a species. So based on this reasoning, Grauer concluded that at most 10% of our genome could be functional, that is, under selection. However, there are at least two flaws or limitations to this reasoning. First, if functions exist for a large portion of the genome that don't depend on specific sequences, then the genetic load argument doesn't apply. For example, if some stretches of DNA function as spacers, or if sections of DNA specify RNAs with simple shapes that matter, then specific sequences aren't required. A second flaw in the reasoning is that we know today that organisms, or even cells, are able in some cases to find different solutions to achieve the same end result when mutations arise, either through redundant genes or through alternative pathways. This characteristic of robustness and multiple pathways to the same end state is expected within a design paradigm, but it's not expected within the viewpoint of a genome formed by random mutation and natural selection. Nevertheless, it's observed to be true. So for both of these reasons, mutations may occur in functional, non-coding DNA sequences without a strong negative impact on fitness. Remember, a key parameter in these calculations is the fraction of mutations that are expected to be harmful if they occur within functional regions of the genome. The value of 10% for harmful mutations comes from the impact of mutations on the protein coding portion of the genome. According to his view, random mutation and natural selection created the entire genome, and so all functional elements in the genome should have similar sensitivities to the deleterious effects of mutations. On the other hand, from the design perspective, protein coding genes and regulatory networks involving non-coding RNAs and transcription factors are completely different designs, and so there's no reason to think that they have similar sensitivities to mutations. In his recent book, Philip Ball, again no advocate of intelligent design, confirmed that it is now known that much of the non-coding fraction of the genome is involved in transcriptional regulation of genes, and that evolution does not pronounce the final word on what can and can't be functional in biology. In support of a new view of biology, wherein some functional elements are not as susceptible to the negative impact of mutations as once thought, Ball refers to experiments that knock out, meaning inactivate or delete, genes known to be functional. In some cases, these knockout experiments produce the bewildering result of little or no effect on the organism, or negative effects that are unrelated to the known function of that gene. This is due to the fact that cells are much more complex than previously thought, and specifically that cells can have highly redundant pathways for conveying signals, or are able to take other compensatory actions further down the line. This topic of redundancy or resiliency is reviewed in an article in the EMBO journal. EMBO stands for European Molecular Biology Organization, which states, gene knockout experiments have shown that protection and adaptive ability are conferred by networks rather than individual genes. Non-coding RNAs are known to be involved in these complex networks. Dennis Noble reviewed Philip Ball's book for the journal Nature. In his review, he cited an example of resiliency from his own area of expertise, the regulation of the rhythm of the heart. He wrote, "The reality is that organisms are extremely robust, and a particular function can often be performed even when key genes are removed. For instance, although the HCN4 gene encodes a protein that acts as the heart's primary pacemaker, the heart retains its rhythm even if the gene is mutated." Further evidence for important functions for non-coding RNAs cited by Ball, independent of the ENCODE project, includes the well-known fact that the number of proteins in an organism does not correlate with complexity, whereas the amount of non-coding DNA does correlate with complexity. For example, humans and the microscopic worm C. elegans have about the same number of coding genes, 20,000. Yet there's an enormous difference in complexity. For example, this slide shows the total number of cells for each. So the number of proteins in an organism clearly does not correlate with complexity. The chart on the bottom of the slide from a publication in 2004 shows that the far greater complexity of humans correlates with the far greater amount of non-coding DNA in humans compared with simpler organisms like the worm. So the amount of non-coding DNA correlates positively with complexity, indicating that it must be functional. Finally, a third evidence for important function in non-coding DNA, cited by Ball and many others, comes from genome-wide association studies, which examine correlations between diseases or traits and genetic variation. Now that we can sequence genomes at an affordable rate, it's possible to sequence large numbers of genomes and study such correlations. In these studies, the vast majority of the single residue mutations, or SNPs, SNPs for single nucleotide polymorphisms, identified as important are in non-protein coding regions, which clearly demonstrates that important function exists in those regions. A recent review article on this topic states, only 5% of disease-associated SNPs are located in gene coding sequences. The remaining 95% of disease-associated SNPs are located in non-coding DNA sequences, which make up 98% of the genome. Disease-associated SNPs are enriched at the millions of gene regulatory elements that are dispersed throughout the non-coding sequences of the genome, suggesting they function as gene regulation variants. Moving on from the controversy over the original results of the ENCODE project, much has been learned about the function within non-coding DNA since 2012. It is now clear that much of the software in cells is carried out by RNAs. Ball quotes from a review article in 2010 that biological complexity has RNA complexity at its core. This review from 2014 describes the rise of regulatory RNAs, including the statement, "Our understanding of the previously hidden and unanticipated world of non-coding RNAs has greatly expanded in the past 2 decades. Indeed, in retrospect, it seems that we may have fundamentally misunderstood the nature of the genetic programming in complex organisms because of the assumption that most genetic information is transacted by proteins." The authors further emphasize the tremendous importance of this regulatory RNA, stating, "Indeed, it seems that RNA is the computational engine of cell biology, developmental biology, brain function, etc. The complexity and interconnectedness of these systems should not be cause for concern, but rather the motivation for exploring the vast unknown universe of RNA regulation, without which we will not understand biology." In 2021, this paper in Nature reported an explosion in the discovery of genetic elements in the non-coding regions previously called junk DNA, since the completion of the Human Genome Project. The graph shows the number of protein-coding genes in blue and the number of non-coding genetic elements in red. The graph covers the time up to 2017, and at the end of 2017, there were more than 10 times the number of genetic elements in the non-coding regions compared with the protein-coding genes. The explosion has continued since 2017. This article in 2021 included the striking statement that the days of junk DNA are over. A series of papers providing an update of the ENCODE results were published in 2020. This slide shows progress in annotating functional elements from 2012 through 2019. The values are presented as percent of the mappable genome. Roughly speaking, most of the annotation categories on this chart have at least doubled over this time period. Chromatin structure is highly regulated, and is a very important aspect of transcriptional regulation. In this chart, open chromatin indicates regions of DNA that are accessible for processing and therefore highly likely to contain functional elements. Histone modifications are epigenetic markings that indicate alterations in chromatin structure and are part of the regulatory process. This slide shows a summary of the progress in annotating functional genes. Note that from 2003 through 2019, the number of protein coding genes has remained nearly the same, whereas the number of RNA genes has increased enormously. It's clear from this chart and the chart on the prior slide that a large fraction of the genome is functional. The article states that there's still a lot more annotation remaining, such as annotation of elements that are highly selective for specific cell types or condition-specific states. This is a list of some of the different types of functional non-coding RNAs that are part of the recently discovered universe of RNA regulation that comes from the portion of our genome that used to be called junk. This image is from a recent review article on the mechanisms of RNA regulation. Without going into any detail, I only want to point out the basic facts that, number one, non-coding RNAs act through base pairing with strands of DNA or RNA, and number two, RNAs can form simple shapes that are often important in addition to specific recognition sequences. Non-coding RNAs also act through binding to proteins, including transcription factors. Two examples are cited here. Another recent review article points out that RNAs are increasingly recognized to participate in intricately functioning machinery across almost all levels of biological systems. Finally, the 98% of our genome that used to be called junk is now being mined for RNA-based therapeutics, as highlighted by this recent review article. Currently, only 0.05% of the human genome has been drugged by the currently approved protein-targeted therapeutics. Recall that 95% of disease-associated single-site mutations are located in non-coding DNA sequences, which make up the 98% of the genome that used to be called junk. Prior to the RNA revolution, nearly all therapeutics targeted proteins. Now there's a rush to develop RNA-based therapeutics. But we lost at least 20 years due to the junk DNA paradigm. The intelligent design paradigm, which encouraged researchers to look for function in the non-coding portions of the genome, would have led to advances in RNA therapeutics much earlier. This slide shows a small sampling of papers reporting functions for portions of DNA that used to be considered junk. This slide contains a few more references reporting functions for portions of DNA that used to be considered junk. To summarize, this very recent discovery is that the junk DNA paradigm has been shown to be false. A large proportion of junk DNA in our genome was once considered to be an essential prediction if our genome arose by random mutation and natural selection. But not only was this prediction shown to be false, but what has been uncovered within the regions of non-coding DNA is a world of previously unimagined complexity. The results of the ENCODE project and subsequent work have contributed to a revolution in biology, showing that non-coding RNAs play a major role in regulating the transcription of genes. While we don't yet know all the details, it's clear that this newly uncovered world of regulatory RNAs, along with transcription factors and RNA binding proteins, which I haven't talked about yet, form highly complex networks characterized by robustness and redundancy. This constitutes another tremendous informational discontinuity.